In question 5, I'm asking you, how can you figure out that which programs you could use to figure out which is the real ATG as a start codon. So if there's an ATG on DNA sequence, if it is in non-coding DNA, then it's just a nucleotide like any other one, is not special at all. But if it's in coding DNA, then it's like, hey, this is either where the translation starts, or it's a methionine, which is an amino acid, somewhere within the protein sequence. So how could you figure out, when I'm looking at this, there is a bunch of ATGs, which of them is really the start codon? And I did this in class, so I wanted to tell you that there are programs that will look for, okay, if this is an ATG and it is a start codon, then if you look at each tree and you go down the line, I don't think that this TAG is in frame, but meaning if I go each tree here, 3, and then this, oh, okay, these 3. Okay, so when I say that the TAG, which are marked in red, there's a stop codon, the TAG is not in frame, because if I read these 3 here, then TAG doesn't get read together. The ATA is what gets read together. So there are programs that will look, is if this is read as these 3, would this TAG be read in frame? And if so, is the distance between the start codon and the stop codon something that it's reasonable for the length of a protein? And the program also looks, is there a Tata box or other signals? I only told you about Tata box, but there's cat box, there's other stuff. What the computer is going to look for is, are there DNA sequences upstream of that ATG that are part of a promoter or part of a regulatory sequence that will signal RNA polymerase and transcription factors to bind here, and at some point downstream start transcription. So the mRNA actually has some chunk that does not get translated, and then this is where it starts translation. Okay, so what I told you is what FGenes-H does. So it's looking for sequences here to predict where are the genes in the sequence. But you could use other ones too. Let's think, if I did BLAST10, that means I give a nucleotide sequence and the computer is looking for a matching nucleotide sequence. So this one actually is a tricky one. If I put the genomic sequence, which is, I have some DNA sequence that I don't know if it encodes any genes. Maybe that's what I'm trying to find out. And then I search the nucleotide database. If my hit, if the sequence that looks like mine is a DNA sequence that is also genomic, that gives me no clue whether or not there's a gene in that genomic sequence. Maybe it's all non-coding, or maybe it's all non-genic. So it's not any type of DNA that will be transcribed. But if I narrow down my nucleotide database to EST, for example, then I would get hits between my nucleotide sequence that I entered, that could be like this thing, and a list of mRNAs that people entered. So if somebody sequenced mRNA and they put it in the nucleotide database as mRNA, then I have a clue of which part of this matches some mRNA that somebody else has already sequenced. So because I didn't specify that nucleotide here is the EST library, I would not check that. Okay, BLAST-P. If I have this sequence here and I'm trying to figure out which ATGs are my possible start codon, which one could be the real one, Then what I do have is nucleotide sequence. And so I would not use BLAST-P because BLAST-P you have to have protein sequence and you're comparing to a protein database. But if you have nucleotide sequence, then don't pick BLAST-P because you don't have protein, you have nucleotide. All right, so the last one, you have a nucleotide sequence, and then the computer would translate your nucleotide sequence to 6 possible frames. So that's what is meant by translated nucleotide. You're only entering the nucleotide, the computer is doing the work of making the 6 possible ones. And then it compares each of those 6 protein sequences that are all virtual, you don't know if any of them exists, compares to a protein database, which is DNA sequence, or it is protein sequence that we know exists. So somebody else sequenced the protein. I know the amino acid sequence of that protein. So it's deposited on this bank of protein sequence. So if there is a match between at least one of my 6 translated nucleotide sequences, if there's a match between that and the protein sequence, then I could see where which of the ATGs is the possible or the likely start codon. So I would mark that too. You will do another BLAST exercise where you are actually doing this.